learners. Welcome to today's Life Sciences Paper 2 revision lesson. My name is Fawzi Rahman, and this lesson is brought to you by the Gauteng Department of Education in collaboration with Saibono Discovery Center. We are going to move on with Paper 2 revision. And this is all the problems that you had in the Gauteng prelim Paper 2. That's what we're going to be discussing today. So that's the revision program. Let's get started. The content weightings in paper 2, as you can see, you have DNA, which is a large amount of marks, genetics, which is 48, and evolution, which makes up 1/3 of the paper with 54 marks. Meiosis is a little smaller amount of marks, but a very problematic section, and that was problematic in paper 2, and that's what we look at now. So meiosis was 21 marks. We're going to look at, these are the concepts we're going to look at in meiosis. In cell division, where a diploid cell undergoes 2 cell divisions to form haploid gametes. That occurs before DNA replication, and then there's a first division, a second division, and the biological importance is what you got to make sure that you go back to exam guidelines and learn. The terminology, this is a list of terminology that you must know for meiosis. And you know, terminology is asked in question 1.2, but with life sciences, it's all about terminology. And to answer the entire paper, you've got to understand your terminology. So make sure you understand this, write it out, and learn the difficult words that are very similar but different. So starting with meiosis, there is the first phase that occurs is interphase, during which DNA replication occurs, right? And each chromosome now consists of 2 chromatids joined together by a centromere here. This is when DNA replication occurs, right? So what happens is, what does DNA replication do? It helps to double the genetic material to be shared equally between the cells. And this is the process of DNA replication on your right that you can see. Now, how do we identify the phases in meiosis? So what we've done here, we've put the first meiotic division and the second meiotic division, and I'm going to try and explain to you how you can look at and work out the difference between both the phases, because you are asked this in the exams and many of you are not aware of how to differentiate between these 2 phases. So firstly, we look at prophase 1. Prophase 1, remember P for prophase, P for preparation for cell division. And in prophase 1, you can see that there are homologous pairs of chromosomes that come together, and this is the stage where they are known as bivalents. It's only at this stage you can call them bivalents. A very important process occurs in prophase 1, and that is crossing over. And remember, crossing over leads to variation. If you look at prophase 2, you can see that the chromosome number has already halved, and you have 2 chromosomes here with the nuclear membrane and the nucleolus have disintegrated, and no crossing over occurs in prophase 2. You can see 1, 2, 3, 4 chromosomes there and 2 here in each cell. So that's how you differentiate for prophase. Let's look at the next phase, metaphase. Here you can see that the pairs of chromosomes align at the equator. They align randomly, and because they are aligned randomly, that is the next source of variation. So M for middle, M for metaphase. If you look at metaphase 2, they are no longer homologous pairs that are arranged at the equator. It's single chromosomes that are arranged at the equator. So chromosomes align singly, and that's the word that we're going to look for at the equator. And because they also align themselves randomly, that is the next source of variation. So metaphase 1, metaphase 2, and remember prophase 1 all give rise to variation. The next phase is now anaphase. A for anaphase, A for apart, because here the chromosomes are going to move apart. So in anaphase 1, you can see that chromosomes move, the spindle fibers contract, right? These are the spindle fibers here. The spindle fibers contract, and that's how the chromosomes are pulled apart to opposite poles. Remember that if they fail to separate, then this leads to a very important process called non-disjunction, and this generally occurs in with the chromosome 21 pair, and then we give that name that leads to the disorder called Down syndrome. With regards to anaphase 2, now remember here you've got homologous pairs that are aligned at the equator, and one whole chromosome is pulled to the north pole and one to the south. But in anaphase 2, because we have a single chromosome that is being pulled apart, the centromere splits. And that's how a chromatid moves to the top and the other one moves to the bottom. And you can see generally they are sort of bent chromosomes because they seem to be pulled to opposite ends. Telophase, T for telophase, T4, two cells form, the nuclear membrane reforms, the cell membrane constricts here, and cytokinesis occurs where the cytoplasm is divided. Now each of these cells will undergo cell division to form two cells, and that's how we end up with 4 cells. So each daughter has half the chromosome number, 4 haploid daughter cells form, and an important point to note is that each of these cells are different because the chromosomes have exchanged genetic material, so each of these chromosomes are now different. So we have 4 non-identical cells or genetically different cells. We're going to take a look at the question, in particular the 2.3 in the Gauteng prelim exam that was very, very problematic, right? It showed you the diagram represents cell P in a phase of meiosis. So it just says a cell, not in particular an animal cell, it's just a cell. And you can see that, let's unpack this diagram, okay? You can see that there is a chromosome, there's spindle threads, there's the centrioles on either end. Okay, so we've looked at the diagram, and let's see now what's the question. Name the phase of meiosis represented in the diagram. So you can see that the chromosomes are aligned at the equator, so they're in the middle, it must be metaphase. But now your answer will not be complete if you just say metaphase. You've got to give the phase of meiosis or the division of meiosis, either 1 or 2, to get your answer correct. If you look at this, it's a single chromosome. So this must be metaphase 2, right? Let's see if we are correct. It's metaphase 2. The next question, give the number of chromosomes that were present in the parent cell of P. Because this is the second meiotic division, and we know that by the end of the first meiotic division, the chromosome numbers half already. Therefore, there must have been 4 in the original cell. 1 daughter cell at the end of meiosis in cell P, and that is also going to be 2, because remember we've got 2, there's 1 and there's 2. So at the end of this process, we're still going to have 2 chromosomes because the chromosome numbers are halved. We had 4 and now we're going to have 2. Give the name of the structure A. So what is A called? A has 2 chromatids joined together by a centromere. That must be a chromosome. Okay, give one function of structure B. You're always going to get this. Please make sure you understand that B is the spindle fibers or spindle thread, you can say. And the function of that is to contract and pull the chromosomes or the chromatids to opposite poles. Describe the process of crossing over that resulted in the appearance of structure A. So let's look at structure A. Each one of these chromatids that are aligned here are different. And the process that occurred to make them different happened in prophase 1. Remember, we spoke about that crossing over, and that is for variation. So let's describe that process that occurs, right? It happens during prophase 1, that's 1 mark. Homologous chromosomes lie next to each other. Chromatids from each homologous chromosome, or you could say non-sister chromatids, overlap and touch each other at a point called the chiasma. Please learn this, and then chromatids exchange genetic material. So if you look at the diagram, it helps you to understand that these are the non-sister chromatids exchanging, overlapping and exchanging genetic material, and afterwards you see that the chromosome has a bit of the different color, so it is a new chromosome now with genetic material from the other pair, and that's why this now leads to variation. So this is crossing over. The next question says, draw a diagram of the phase that occurs after the phase in the diagram. So remember, we are at metaphase 2. After that will be anaphase 2. So let's look at how we're going to draw that. Now, this is the phase, this is the diagram, the original question. All you have to do is copy from your question and show them moving apart. So they must be on the same side. If this is on this side, then this one here must be on that side. That one is going up. The spindle fibers have contracted and it's pulling that chromatid up. It's pulling this one here down this way. And then you've got this one, which is represented there. And it's a smaller one. You can see these are bigger compared to this one. So if you draw these accordingly, you just have to show the separation, then you're going to get your marks, right? So I just want to show you the rubric here. You must have a caption to say that this is a diagram. You're drawing a diagram of a cell in anaphase 2, because remember the first phase was metaphase 2. You must have a caption, your position of your chromosomes, large one, a smaller one. The shading must be correct, and any two correct labels. So you can label the cytoplasm, you can label the spindle thread, you can label the centrioles. So learn at least 3 labels that you can use in any diagram that you are asked, and with your caption you can get quite a bit of marks from the diagram itself. We're going to move on now to genetics, and this was another problematic question in the Gauteng prelim paper 2 exam. Remember, genetics is 48 marks, and these are all the different concepts that you need to learn for genetics. We are going to look at a few of them that were problematic, as I said. Right, the terminology. Concepts commonly used and confused and used interchangeably but are incorrect to be used interchangeably. For example, there is one gene for a particular characteristic, maybe color. There'll be 2 different forms of that gene. So 2 different alleles, a red allele, a white allele. In the same way, dominant and recessive, phenotype, physical appearance, genotype, genetic material, homozygous and heterozygous. Make sure you understand the difference between these and you are able to explain them. Now we're going to look at Mendel's law of inheritance, and if you look at the exam guidelines, page 11, page 12, you will find these and you have to learn them. They are going to come out in the exam. You must be able to study them and write them out. So then you know for sure, you understand and you know the definition, not just by reading it and learning it, but by writing it out. One of these will definitely come out in your exam, so make sure you learn these. That's easy marks to score. Now we're going to look at the format of a genetic cross. And this is critical. In order for you to get your answer correct, you have to follow these steps in order to get your mark and to get to the correct answer. Right? So the layout of the diagram, the first one is the phenotype, is P1. P1 tells you that it's the parents, the original parents. Then you write down the phenotype, that is the physical appearance, right? Tallness, shortness, curly tail, straight tail, curly hair, straight hair, whatever it is, that's the characteristic. Then you look at the next one, which is the genotype, and you get a mark for the genotype, which is the genetic makeup, which you get from the phenotype. And then you have the process of meiosis occurring, where you get your gametes forming, and then fertilization occurs, and then you get your first filial generation or your F1 generation, and then from your fertilization, you will get your offspring, which you can write then as your genotype and work out your phenotypes. Now this P1 and F1 that's written here, that's not where you're supposed to write it. That's just for the marker to see, to give you the mark if you have it in the correct place. So if we look at, so So the alleles segregate to form the gametes, and during fertilization, each one gets one gamete from each parent, and that's how you work out the genotype there. So make sure that you write the P1 and the F1 in the correct place. Also, meiosis must be before gametes, because remember, meiosis occurs and then your gametes form and then fertilization occurs. So they must be in the correct place to get your 2 marks, and that is easy 2 marks to get if you just follow the format of a genetic cross. Now, what are the steps that you need to follow when answering a monohybrid cross? Remember, monohybrid, mono means a single characteristic, right? So the first one is identify the phenotype of the parent. Then you will choose the letters to represent those alleles. In most times they are given to you. If they are not given to you, you will use the dominant characteristic, and you'll give that the capital, for example, T, if T is tallness and that is dominant, and then the allele, the opposite allele for shortness, you'll use the same letter but the lower form, and that would be the small letter T. Identify the genotypes of the parents, show how the alleles are separated through meiosis into separate gametes, and show all the possible genotypes of F1 generation when fertilization occurs, and then you interpret the phenotypes. Remember, you got to answer the question. If the question says give the probability of, that will be your compulsory mark. You must have that in your answer in order to get full marks. Now, genetic crosses involving only a single characteristic is called a monohybrid cross, mono, single. You have complete dominance, incomplete, co, sex-linked, blood groups, and the dihybrid cross, which involves 2 characteristics. That's why the word di. Let's look at complete dominance, right? A genetic interaction when one allele of a gene suppresses the expression of the alternate allele, right? In the F1 heterozygous state. So if you look here, red is dominant over white. How do I know that? Because when I have a capital R and a small r, I still get the color red. The only time I will get a white flower is when I have a small letter r and a small letter r together, the recessive, homozygous recessive state. As long as I have the big R and the small r, the color of the flower is going to be red, because red is completely dominant over white. If I look at incomplete dominance, here neither of the alleles are dominant over each other. So an intermediate color forms. Now, this is the notation. It's capital R with capital W, and then capital R and capital W. But the color will tell you what kind of cross this is. So it's incomplete dominance because the offspring that forms is a blending, is a mixture of both the parents. Red and white give you pink. And it's the second way you can find out why it is incomplete dominance, because the pink flower is it's a new color compared to what the parents wear. It's a new color forms, and this new color is a blending. So if you have a black rabbit and a white rabbit, and if they mate and form a gray rabbit, that's incomplete dominance as well. Now we look at codominance, and the notation here is the same, but how will you know that this is codominance is because of the color of the offspring. In this case, both alleles are equally dominant and they are both expressed in the offspring. So the red is saying I am dominant, the white is saying I'm also dominant, so the offspring will have red and white. So you can see the red and white flower here, and this is codominant. And an example, you may get a black dog and a white dog, and you'll get a spotted dog like the Dalmatian, and that is also co-dominant. And majority of the time you get questions in relation to co-dominance, and you find them when we do blood groups. So we're going to do that just now, so have a look, a mental note of that. Okay, so we move on to sex determination. And these are karyotype that's showing you the 20 from number 1 to number 22, the autosomes. Those are called autosomes, right? And generally we study genes or disorders in genetics that are autosomal. But you can also get diseases that are found in your gonosomes or your 6 chromosomes, and your 6 chromosomes are found on the chromosome number 23, your 23rd pair. That is called the 6 chromosomes, or a less confusing term to use is the gonosome. So these now are your gonosomes, right? So that's the chromosome number 23. Now let's look at sex-linked disorders. Any disorder that is found in the gonosomes is called a sex-linked disorder. And these genes are located on a 6 chromosome. They are called sex-linked genes. In human, the term usually refers to X-linked characteristics or genes located on the X chromosomes. Now, fathers easily pass on their X alleles to their daughters, because remember, a daughter is X, and she gets one X from her mother, one X from her father. But mothers pass on their sex-linked alleles to both sons and daughters, because the son must get an X. Who he gets it from? His mother. The Y he will get from his dad. Okay? Now, what are sex-linked traits? These are traits controlled by genes located on the 6 chromosomes. Guys, when you hear the word sex-linked, that is the only time you will use the notation X and X for a female, X and Y for a male. There's no other time that you will use this notation until you are told it is sex-linked. So when you see in your question while you are reading during your reading time, or when you are starting to answer the question, you highlight, you underline, a bell must ring in your head, tilling, sex-linked. What is the notation I need to use? X and X for a female, X and Y for a male. Excuse me. Most of these traits are X-linked. The Y chromosome, as you can see here, is a tiny chromosome. There are not many genes on that chromosome. So we find that if there are any disorders, you find them on the more genes are found on the X chromosome. The Y is tiny, right? And please note that there are no alleles on the Y chromosome. The Y is written on its own, just a Y. But the X will have a superscript H. So if this is showing you the disease for hemophilia, for example, and that is found in the recessive state, then this individual will not have it. This individual will not have it. This one will have it. This is a male and he will have hemophilia. This is a female. She will not have it because she has this dominant H to mask the effect of the hemophiliac gene on this side. Okay, so let's look at what I'm saying. Males are more affected by recessive X-linked traits. Why? Because they only have one X chromosome and they do not have the second X to mask the effect of that. So here, if you find D is dominant, the dominant allele will be expressed there for the female, for the male here. And in here, the female will have the recessive allele expressed, and here for the male, he will have the recessive allele expressed there as well. Okay, so I hope you clarified that in your minds, and we're now going to move on to blood groups. I know you do not have a question on blood groups in the prelim exam, but you will get one in your final exam probably, and it's nice to just iron out the creases for this. The inheritance of blood is an example of multiple alleles. Why we say multiple alleles? Because you have an I superscript A, I superscript B, and for the group O, you have a small I. The notation for the alleles indicating blood groups is I superscript A, so I'm going to say IA, you understand what I'm saying, IB, and I. Different combinations of the alleles arise or result in 4 blood groups. So we have blood group A, blood group B, blood group AB, and blood group O. It's important for you to say blood group A or write it down in the exam. And the difficulty with blood groups is that because the phenotype and the genotype are both letters, it makes it difficult for the learner to understand. So because your phenotype is a letter, your genotype is also a letter, so that is a confusing part about blood groups. So make sure you understand that A, B, and O, those are the phenotypes, and you must use the words blood group A, blood group B, blood group AB, and blood group O. The alleles now for the genotypes. I superscript A and I superscript A is for blood group A, and remember O is recessive, so the small I there will mean that A is dominant over O, so we'll have blood group A, here blood group B. This is what I was talking about, the co-dominance. Because A and B are co-dominant, your antigens on your blood cell will be A antigens and B antigens you will find on your red blood cell. Here you have the A antigen, here the B, here you'll have both A and B. So they are showing in your blood cell. That's why both are dominant, they are co-dominant. In your blood group O, it's there are no antigens, right? And majority of us are blood group O. That's completely dominant. This is completely dominant. But when you come to A, B, we come to co-dominance, and then we have complete dominance again. So you need to learn the 1, 2, 3, 4 rule of blood. It's one individual with 2 alleles in that blood group. Remember, you get 1 allele from your mom and 1 allele from your dad. There are 3 different alleles controlling blood, IA, IB, and small I, and there are 4 blood groups, blood group A, blood group B, blood group O, and blood group AB, which is a co-dominant one. Right, that's a wrap with blood. We're going to move on to genetic pedigree diagrams or genetic lineages. Now, this was a very big problem in the exam. So let's see, a genetic pedigree traces the inheritance of characteristics over many generations. And when you see a pedigree diagram, because we are looking at male and female, it does not mean that you must use X and Y notation. X and Y notation is only for sex-linked characteristics or diseases or disorders. It is not all pedigree diagrams that you must use X and Y. It could be you're given a pedigree diagram that is related to blood, for example, right? So then you will use the notation IA, IB, and small I for the O group. So this is just to trace any disorder that they may be or any characteristic that they may be through your generation, your mother, your grandfather, great-grandfather, that type of thing. It is not that when you see a pedigree, you must start using X and Y. No, that was the biggest problem for this example in the prelim that we had. Everybody saw a pedigree and they use X and Y notation, but nowhere in the question did it say sex-linked. So let's look at how you're going to know the meaning of this. Firstly, you look at the key or the legend here, and a circle is generally a female, a square is a male. If the male is shaded, then that is the one generally that is affected, and the female that is shaded is generally the one that is affected. A horizontal line between a male and a female shows partnership, and then you get a line of descent. Right, that's the offspring, and this bracket just shows you that these two here are siblings. The first line shows you the first generation, the second line shows you the second generation. Okay, so know the meaning of these symbols and the lines in these diagrams. The next one is the step involving your pedigree diagrams. You need to study the key. Firstly, study the key. Look at male, female, shaded, unshaded, what does it mean? And then you look for the dominant and the recessive characteristics, and you fill in the genotypes of all the individuals with the recessive condition. So you will look at your diagram, go to your diagram, and fill in on your question paper all the small letters, the homozygous, recessive, fill that in on your diagram. And remember, to find out about the parent, how are you you to know what the parent is, you have to look at the offspring. The offspring tells you what the parent is. So if the offspring is recessive, small t, for example, it must get one small t from the mother, one from the father. But if the mother and father are showing the dominant characteristic, then they are heterozygous. Then you'll put a capital letter next to the parent, okay? So that's how you will work out the pedigree diagram. We're going to look at an example from the Gauteng Prelim Paper 2, question 2.4, because this was very, very poorly answered. Familial hypercholesterolemia, or FH is a symbol we're going to use, is an inherited condition caused by a dominant allele. Highlight all the important words. People with this condition have high levels of cholesterol in their blood. This increases the risk of dying from blocked arteries. The diagram alongside shows FH inheritance, the pattern in several generations of a family with this disease. And we are telling you dominant allele is big T, recessive allele is small t, but majority of you went and used X and Y. That's the question. Nowhere in the question did it say sex linked. So do not use X and X for a female, X and Y if you are not told that it is sex linked. You do have male and female here, but we are giving you a situation where we have complete dominance, therefore a big T and a small t. Right, so let's unpack this diagram. We can see that we have a square, which is a male, and that is shaded. So this male here is the male with the disease, and the male without is unshaded. The female is unshaded, she is without the disease, and the male then has the condition, right? Then we have 1, 2, 3, 4 generations, okay? And then we have the children of A, B, and the whole family line there. So let's see what the question says. How many generations are shown in the diagram above? You can see that's 1, 2, 3, and 4. So should be 4, right? Determine the number of males in the diagram whom are homozygous recessive. If you had put already looking at the key and you went and you wrote down the homozygous individuals, you'll have your answer already. So let's see. Homozygous recessive will be the male without the condition, and we are looking for males, therefore we'll be looking for squares that are unshaded. So there we have 1 there, that's D. We have M, and we have R here. So we should have 3. Right, couple G and H. So we're looking at these two. Couple G and couple H are planning to have a 4th child. Let's look at their children. Their children are N, O, and P. They are planning to have a 4th child. Use a genetic cross to show the percentage probability of having a child without FH. So we are looking for a child that is unshaded, because they have 3 that are shaded now who have the condition. Now, if you look at G and H, right, they are coming from parents C and D. G is coming from parents C and D. Remember, one allele must come from the father and one allele must come from the mother. The father is small t, homozygous recessive. Therefore, G will have to have a small t. But G is showing the dominant characteristic. Therefore, G must be big T, small t. And H, obviously, she married a person who does not have the condition. So this person will be small t and small t. Now, when we do the cross, you can see we've got a big T, small t, and 2 small t's, 1 mark for that. Then you will show meiosis occurring, which should have been a little bit at the top. And then there's your gametes. We will take one of the individuals and write them in a table horizontally at the top, and we will put the second one here vertically. And then you show fertilization or a cross. Big T and small t give you that, small t and small t there, small t and small t. So when we write this out again, we have big T, small t, 2 here, and we have 2 there. So that means that the probability without the disease is homozygous recessive, and that will be 50% of the individuals, right? They'll have a 50% chance of having a child without FH. And that is answering the question, therefore that would be the compulsory mark, and only if you have the compulsory mark will you be given a total of 6, because it's a compulsory mark plus 5. We now move on to the dihybrid cross. This is a cross where there are two traits that are crossed, one individual, and there are two individuals that are crossed, but you are looking at two characteristics. So you are looking at short and long, or you're looking at curly and straight, and you're looking at color, black and white. So two different characteristics, right? So two traits are crossed, and the word di should tell you dihybrid. The law that we look at for this is Mendel's law of independent assortment, and you need to read this and understand that law. And remember, the genes for these are on different chromosomes. So when you cross two heterozygous organisms, you find that the phenotype is in the ratio of 9:3:3:1. And in the exam, when they ask you probabilities about this question, you can work out easily by saying 9/16 and work out the probability depending on what question they ask you, the dominant and dominant, or dominant and the recessive. You can easily work out the percentages without doing an entire genetics cross, because that would be too long and a waste of time. So let's look at the last question, question 1.5 in the prelim. Cats inherit many genes that affect the appearance of their coat. One gene controls whether the coat is striped or a solid color. So now we're looking at color, right? The agouti allele A, and we are giving you that is capital A, for striped fur is dominant over the allele for a solid color. So here we are talking about two alleles, color, and the second gene affects the length of the coat. So the coat is either long hair or short hair. So can you see we are looking at two different characteristics? Therefore, this is a dihybrid cross. Agouti allele for striped fur, striped is dominant over colored, a solid color, and long is dominant over short. Therefore, capital L for long, and short is a lowercase. A male who is heterozygous for both agouti and long hair, right, is mated with a short-haired, solid color-haired female. So if he is heterozygous, remember he's showing the dominant characteristics, but we are saying he is heterozygous, therefore he will have let's answer the question first. The type of cross is a dihybrid cross, we said that, right? The genotype of the female cat, remember she is short-haired and solid-colored. Therefore, she has both of the recessive traits. So all her alleles will be the small letter. Now, big A is for striped, therefore small a will be for solid. Long-haired is capital L, therefore short-haired will be the lowercase l. So you can write them A-A-L-L, or you can put them the opposite way, L-L-A-A. You're still going to get the mark. So now comes the question that I was trying to answer in the beginning. A male who is heterozygous for both agouti and long hair, he mates with a short-haired, solid-colored female. So remember, A is dominant, and we are saying he's heterozygous. Therefore, he will be big A. Now, you need to understand that we need to give the possible gametes for the male cat. So what you have to do is you have to for every characteristic, there are two alleles. So you keep them separately, and that's the way to answer a dihybrid cross. Keep them separately. Keep the color separate, keep the other characteristic, the length of hair, separate. So now we know that he is agouti, so he will be big A and a small a, right? Because that now talks to the color, and that is striped. And then because he's long hair but he's heterozygous, he'll have a capital L and a small l. So this is the genotype of the male cat. But the question is saying possible gametes. So we need to work out the gametes for this cat, right? So he will have A, L, A, capital L, capital A, and small a, capital L, and small a, small l. Okay, and that's where you'll get the 2 marks. These are the genotypes for the male, or you could write it in the other way, that's fine. Right, and if you look here now, these are the gametes, all the different gametes of the male. The female, these are her gametes there. Okay, because remember, she is a short-haired female, solid colored haired, what that she has. So the inheritance shown by both here is complete dominance, because the striped is completely dominant over the solid color, the long hair is completely dominant over the short color. What percentage of offspring will inherit the same phenotype as their mother? Right? And the answer for that is 25%, because if you put the mother, hers is AL. Remember we said that, so her gamete will be AL, and then there's the father's gametes over here, and if we cross them, we get that one, that one, that one, and this is the mother, and there's 1 out of 4 that is like the mother, so that will give you the 25%. Because percent is in the question, 25 gets both the marks. So I hope you understood and ironed out your creases for these topics that were problematic. And that's a wrap from me. So thank you. Please stay right where you are. Next up is Dr. Griselda van Wyk, who will take you through evolution.
Good afternoon. Welcome back. I'm Griselda van Wyk and I will take you through evolution. But like the previous presenter said, we are only concentrating on the prelim papers today. And what did you do wrong there? Right, so we're trying to mop up and see if we can help you. Right, so firstly, I'm going to just touch on the key concepts that you missed, or where a question was based on and you didn't understand. So, evidence for evolution, remember it's fossils. You know what a fossil is. Biogeography, this is the one where they'll usually also ask some speciation. If you can look at this one, Earth was one big fat continent, and then the continent started to move apart, but there were animals on these continents, and there was common ancestors, and as these continents moved away, they started with speciation and changing to different species. Genetics is the third one, right? So this is the third base of evidence. So when they talk about amino acids, DNA, genes, mutations, all those type of things, you know that that's genetic evidence. They are referring to genetic evidence. And homologous structures are like, for example, the most famous one is the arm of our arm, for example. If you compare it to a cat, a dog, a bat, a whale, you'll see that it's exactly the same bones, but the bones are now different. Look at this one here and compare it to this one there, and it changed through the years. And a bat, the fingers of a bat is now part of its wings, right? The whale is part of its flippers, and that's a cat. There's where their fingers are now, part of their toes, right? So it's the same structures, but it just looks different now, right? It has modified, right? So those are the four evidences for evolution. Now we get to phylogenetic trees. People, you need to understand how a phylogenetic tree works. Remember, they can give you anything. They can give you one that is vertical, they can give you one that's this, and they can put anything on it, and you must be able to read it and understand it. So the first thing that you must look at when you look at a phylogenetic tree is, where's the timeline? Right, it's their timeline. Because that's important. Usually they will ask some, you have to read the time that this one originated, or how long did this species live for, or whatever, right? So timeline is very important. Secondly is your ruler, right? Remember what I always say, your finger is not straight. You maybe think it is, it isn't. You use a ruler, right? Now, quick things with a phylogenetic tree. You will see that this is the root part. This is where your common ancestor is. These are the descendants from that common ancestor. This is speciation event, right? This is a speciation event. A new species has formed, and that is extinction. When the line stops, it's extinction. They are not there anymore. Now you will get fossils, right? And in this one, you can see 0 is there. That's present time. So here you can see are the two species in this case that's still alive. It depends on which organisms they give you, which phylogenetic trees they give you. They can give you basically anything, but if you understand how to read it, it helps you. So make sure that you look at all the different papers, at all the different phylogenetic trees throughout the years, that you understand those concepts. Right. Now let's look how they asked it in the prelim paper too, because this was a problem. Study the phylogenetic tree showing the evolutionary relationships between birds, right? So they didn't give humans, birds, aves, and reptiles. I'm not going to pronounce that name there. And answer the following questions. When did aves or birds branch from the crocodile or the reptiles? Right. So now there's a timeline right there. And now we look at our tree. You see there it starts. Can you see? Because this is the present. This is millions of years ago. So here there was a common ancestor, and it's split into two. Here's the birds. There it is. You don't even have to know what it means. There's aves, they said aves, and there's crocodilidae. There it is. So they split here. So what are you going to do? You're going to put down your ruler, right, not your finger, and you're going to just measure it down here. Right, and then you will see it is 250 million years ago. You have to say ago, million years ago. Right. If they ask you how long it's been alive for, and you have to measure this, then you can't say million years ago, then you will say million years. Then you measure, like, for example, 70 and whatever there, then it will be 70 million years. That one was alive, or that species were around for 70 million years. But if they ask you how long ago they lived, or how long ago did this happen, then you have to say million years ago. Right. Unfortunately, in this paper, there was an error at the printers. I think your teacher told you, and this timeline was not printed. And that's why we gave you these 2 marks. Mahala, for free. The paper actually counted out of 148. Right, give a term that describes the common structures shown by a asterisk symbol in the forelin that was inherited from the common ancestor. Look here. Here's the forelin. And now they're talking about 5 types of bones there. And now you can see all the different colors there. And you can see it's the same thing. But what did we just say? Homologous structures. That's it. It's just, it's the forelimbs. But now they are doing different structures that were modified. Right, homologous structures. That was a very easy question, actually. Right, now we get to the theories of evolution, Lamarck and Darwin. Well, I must say the feedback that I got from all your teachers were you didn't do well in this question. Not at all. You guys were struggling with these questions. And actually, people, it's so easy. But let's just go through the theory very quickly. Law of use and disuse means he said, Lamarck said, and remember this is way back in the 1700s, very long ago. They didn't know a lot there back then. He said he was watching these giraffes and said, oh, you know what, that one, the tree is high there, the leaves are high there, and this giraffe is hungry, so now he's stretching its neck, stretching, and while it's stretching, the neck is actually growing. Imagine that, eh? So now the neck is growing, it's getting taller and taller, and that is law of use and disuse. The more you use it, the longer or stronger it gets, right? Which is not true, we know that. And then inheritance of acquired characteristics says that now this giraffe's baby, if you stretch your neck, your baby will be born with that stretched neck. That is what that law says. So the giraffe's babies were all born with those longer necks because the parents were stretching their necks, which is also nonsense because if you think about it, if you get into a car crash, you lose your leg. Are your babies all going to be born without legs? No, of course not. Can't be. So that is what he said. Because remember, the change is in the phenotype, not in the genotype. Right. And that's why we reject Lamarck. But Darwin, what he did is he said, he talked about the theory of natural selection, right? And he said that all species were not created like they are now. And if we look at the whale, look at the whale there. It was like a dog thing. You can see there, it looked like a hyena dog thingy running around. And obviously things changed while this hyena thingy species were, for millions of years, were running around there. And all of a sudden, maybe it became more water and more water. So they started to go into the water more. You can see they started swimming for their prey, for food, and so on, right? And millions and millions of years later, obviously it was more better for the ones that were able to swim better to survive, and they reproduced more. And you can see how their forms changed throughout the years. And now from that dog-like thing to a whale. Can you believe it? Right, so that's what he said, and he also proposed natural selection. Now, let's look at our question where you did very badly, unfortunately. But let's fix this for the finals. Now, I don't want you to make exactly the same mistakes in the finals again. They're not going to ask this question in the finals, but I just want you to get what we are telling you to do. Please do as you are told today. Right, so this is a swordfish. They are telling you it's a swordfish because it's got like the sword-like nose. Ancestors of the swordfish had short swords. Modern swordfish had long swords. Swordfish use their swords to injure prey. They actually stab them with that. And then they can catch them and eat them. Right, so it's like a weapon they have. The diagram below shows one theory of how the length of the sword of the swordfish evolved. Now, let's read this theory. The sword grew longer, and I hope you did read this. I hope you didn't just glance at it and didn't read this, because if you didn't read it, you couldn't answer the question correctly. Don't be lazy. The sword grew longer as each swordfish used its sword more and more. Now, don't know who said that, Darwin or Lamarck. Who said if you use something more, it will become longer and longer or stronger and stronger? Yes, you're right, Lamarck did, nay. Each time the swordfish reproduced, the longer sword was passed on to the offspring. And you can see here, as he used it more, the swordfish's sword became longer. Right, so obviously that's a Lamarckian statement, right? So, which scientist suggested the theory shown in diagram 2? Just said the answer, nay. Lamarck. Darwin definitely didn't say that. Right. Explain one reason why this theory put forward by the scientists in question 3.21, this first question here, was not accepted or is not accepted. Well, firstly, I mean, obviously, if you use it more and more, it's not going to grow longer. Right. That is obvious, right? So the acquired characteristic of the sword size cannot be inherited. Oh yeah, that's the second law. Acquired characteristics cannot be inherited. Remember the word acquired. Acquired means something that you get while you are alive, right? So it can't be inherited because they are not part of the genotype. Remember what I said, it's if you chop off your arm, it's your phenotype that changes. Your genes are still the same. Right, so when you have a baby, the baby will inherit your genes, not your phenotype. Right, so that's why it cannot be inherited. And a constant use of the swordfish will not make it increase in size. Obviously, if you use it more, it won't grow longer. That's not possible. Or the swordfish can also not choose to grow its. You can't sit there and think, I want a longer sword, and then boop, 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 there it grows. It can't. It's not Pinocchio. It cannot do that. So it got larger due to environmental pressure. Right, so that's why the mark was rejected. Can you see that they accepted 3 facts? Because it's explained, you had to say the cause. So it cannot be inherited, that's the law. Or constant use of the sword, that's the law of use and disuse. Apply to this scenario, the swordfish, right? And then secondly, you have to explain why, how. How is it possible? How is it not? Because it's not part of the genotype, will not make the sword increase in size, or it got larger due to environmental pressure. Can you see there must be a second part when they ask explain? So there was 3 possible explanations for this question. There was only 2. Then describe how natural selection could result in a modern swordfish having a longer sword than their ancestor. And you know what the teachers told me? They told me you just wrote the generic thing there. Just the generic thing. And people, yes, 10 years ago we accepted that. No longer. No longer. Now you have to apply that recipe to the scenario they give you. In this case, it's a swordfish. So now you must think about the sword. Right. So what does the recipe say when you work with natural selection? In the beginning, there's a lot of variation, right? So, but now you have to say, in what? In what is this? Is it their eye color? Is it their tail length? Is it fat or thin or what? In this case, it's a sword length. That's where the variation is. That's what's making all the difference here. So you have to specify, people. If you didn't specify, you didn't get the mark, right? You cannot write the recipe as is anymore. You have to apply. That was the first thing where you lost your marks. Then, now we have to talk about the favorable characteristics and the unfavorable. But you can't say some are favorable and some are unfavorable. You now have to say what is favorable and what is not. Favorable is the long one, isn't it? Because that's where you can kill the prey. So some had a longer sword, and some had shorter sword. All right, so that's your unfavorable and that's your favorable. Then now you must say what happens to the unfavorable and what happens to the favorable one. Right, so what changed? Because you also have to talk about what changed in the environment. In this case, there was competition for food, or you can say there was less prey or less food. That's why they needed to do something with their noses so that they can hunt better. Right, so the ones with the favorable, the longer sword, were able to capture more prey or injure more prey, right? And they survived. So for the survived, you got at least a mark there, but you had to write it in the correct way. Then while the swordfish with the shorter sword captured or ate less prey or no prey, and they died Remember, this is now the unfavorable one. So you've got your favorable one and your unfavorable one. And the longer ones survived and reproduced, right? And here is to pass the allele, but you have to say what allele. You can't just say the favorable alleles. You have to say for the longer sword to their offspring. And the next generation will therefore have a higher proportion of individuals with a long sword. So can you see how important it is to apply the recipe to the context? You didn't do that. We know. We asked the teachers. And the teachers all said, no, none of my children applied this to the context. And this is where you lose your marks, people. So just when you get that question in the final paper, think, look at the scenario they're giving you. And what is the favorable one? What's the unfavorable one? And then you apply the recipe. Okay, so please don't just write the generic thing there, just the recipe as is. You're not going to get the marks. Right, and this was another one. Okay, the emus and the ostriches are both large flightless birds. Remember, we have a lot of ostriches in South Africa. Emus, we don't have a lot. People do import them, but they're ugly. The ostriches are much nicer because they're South African. Emus are only found in Australian, that's why they're not very nice looking, continent, and ostriches are in Africa. Right, so now they're already giving you the scenario, 2 continents. South Africa, Australia. Right. The original hypothesis, remember hypothesis, not proven yet. Not proven yet. About evolution of emus and ostriches was that they share a common flightless ancestor. Remember what I said? There was one big continent, there was the ancestor there, then the continents were split, and then speciation happened independently. Now I've got these 2 birds that don't look the same anymore. That's what the original hypothesis is. The sea separated the ancestral due to, that's what I just said now. More evidence has resulted in a new hypothesis. New hypothesis still not proven. That the ancestral population of emus and ostriches was a high-flying, smaller bird. Right, so there was an ancestor, it was a smaller bird, and this one could fly. It was able to fly. Right. It could fly long distances and immigrated from Central Asia, so it was flying very far. This one could really fly from Asia to the southern hemisphere, to the southern continent. Right, so this was really a very good flyer. New genetic analysis show that mutations in DNA caused by modern birds to lose their ability to fly eventually. So they actually now also saw in DNA that the mutations that they traced showed them that yes, there was these mutations caused them not to be able to fly. Okay, now, so it was a lot of reading. So you have to underline things here and see what is the key things. This is the key thing. That's stressful one. Now what do they want? They want you to describe how the high-flying smaller bird evolved into different species. Speciation, different species, speciation. Larger flightless birds, the 2 of them on the other continents. Right, so I highlighted this because I wanted you to see that there's only 2 continents. It's very important. If they talk about 3 continents, then in your answer there must be reference to 3 continents. But in this case, there's only 2. Okay. So can you see that I highlighted the keywords? That's what you must do when you get your paper. Highlight or underline those keywords that you can see, or write them next to on your question paper on the rough work, right? That you can see what is the important keywords, right? So now let's start with speciation. A population of smaller flighted birds flew long distance to different continents. So they started there and they flew to these continents. Okay, or we give you a mark for became separated by sea, because what is continents? They are separated by the sea. Okay, so either the different continents or they became separated by the sea. That's the first one. Right. The next one is a generic one. The population was split in 2. That's why I say emus and ostriches. There wasn't a third one. Because I know some previous papers they've talked about 3 populations or 4 populations. In this case, it's only 2. That's the easy mark. There was no gene flow between the 2 populations. Where do you always go wrong here? You guys are talking about 2 species. we're already talking about two species. There's no speciation yet. The speciation is happening down here. Here's where the speciation is happening. You're already running ahead and you're losing marks. You have to talk about population. So remember that word, ne? Population split into two populations, not species. The speciation is coming here now. Since each population being exposed to different environmental conditions, this is a generic one. This is a recipe. So this is a generic mark that you can get if you know the recipe of speciation. Or the selection pressure may be different, or there's DNA mutations. Natural selection occurs independently in each of the two populations. Generic again. If you know the recipe, you get a mark there. These are easy marks and you didn't get it. Such that the individuals of the two populations become very different from each other. Genotypically, phenotypically, people, this is just a recipe. Just what's in the exam guideline. Exactly like that. You just write it down, okay? But the big thing here is you talk about the two populations, right? And just at the end, you will say, no, you don't even say they're species at the end. You just say they won't be able to interbreed again. Okay? So the big thing here is don't talk about species here. It's not two species, it's two populations. Split, and everything is happening in these two populations. Okay, and you'll get your marks there. So this one is easier to get. Where you have to be specific is more in the first line here. Where you have to specify to the context that they gave you. But the rest is very much generic, like the exam guideline. So be happy if you get a question like this in the finals. Right, then you also had a problem with natural, the difference between natural and artificial selection. Apparently that was also a problem. So remember, natural is just what we did now. In the beginning there's variation. Look at these two little butterflies. The one is yellow, the one is brown. Remember, butterflies when they are small or when they are in the first phase, they are caterpillars, right? So there's competition between them because there's a lot of them. So they're competing against each other, right? So then the environmental pressure comes in, and you can see the predators will see the light ones more than they will see the dark ones, the brown ones. So the brown ones are like favored here, right? So they will eat all the light ones, and at the end, through natural selection, there will be more brown butterflies than light butterflies. This is natural selection. The nature is doing all the work. Nature is the selective force, right? No humans, nothing. We have nothing to do with this. But here we have a lot to do. The artificial selection, and we've been doing it for thousands of years, right? But now in the last 100 years, you can see chickens, for example. Look at chickens. Look at the 1957 chicken. Goodness gracious me. That was a thin little thing with no meat on it. No meat on the bones there. So what they did was they took the bigger chicken and they will mate it with a big chicken, and they will get a big offspring. Those offspring will breed with each other, bigger chickens, bigger chicken, mate them, and that's how they were able to get to this. They're doing it with dogs, they're doing it with cats. They're even breeding cats now with small little legs because they are cute, which I think is ridiculous, but anyhow. We did it with plants. People, that's why we have these lovely vegetables today, like cabbage and Brussels sprouts and kale and broccoli and cauliflower. They all came from this one plant, and it doesn't even look like it. They just said, okay, this one has got big fat leaves. Let's pollinate it with itself or with another one with big leaves, and that's how they grew the cabbage, right? So over thousands of years, we actually made like lilies and all those things through selective breeding. Right, so where did you fail now again? Yeah, this question. Flies lay the eggs in the urine and feces that stick on the wool of sheep. The eggs hatch and the larva damage the sheep's skin, leading to infection. So when a sheep urinates or defecates, that sticks to the wool, there's flies, and now it's like maggots here in the skin. Is horrible, right? So what they want, now the farmers are deciding, they are selecting, they're developing sheep with no wool on the abdomen, the legs, the backside, the underside, so that stays clean, so that stuff doesn't stick there and there's no flies, right? So they cross different breeds of sheep to produce offspring with desired characteristics. So the farmers are the selective force here, right? And there's 3 different breeds of sheep. Okay, now using the evidence from the table above, not your own evidence. You don't decide, well, I know this sheep, and I'm on the farm and I'm going to use this sheep. No. This table, that's what you use. It's a very easy question, people. Describe how farmers could use selective breeding, selecting it themselves, right, to get bare legs and bare backsides. Now, all you do is you go and look in the stable and you see, okay, bare head, oh, they don't want that, but they want bare legs. Bare backsides, yes, that's what we want. But there's no bare abdomen, so it's not this one. So we're going to use these two, and we're going to breed and mate them with each other, breed them with each other, and, you know, get that. So cross, mate, breed is the word you could have used. The East Frisian for bare backsides, and the Cheviot for the bare legs. You just said that you got 4 marks out of the 5. Can you believe it? Just saying that, you already get 4 marks. Select the offspring with both bare legs and backsides, and then you interbreed them with each other again, right, for many generations, and then, you know, there you get it. Right, there's many marks given for the answers here. You didn't get it though. Right, so now I want to know, tabulate 2 differences between selective breeding and natural selection. Right, and also you didn't get this. People got confused here with GMOs and stuff. We're not talking about GMOs, we're talking about a farmer in his farm and he's now breeding this one with this one. Right, so what is the differences between them? Firstly, there must be a table, because why? Tabulate. Right, so that's 1 mark, né? 1 mark for the table. Don't lose a mark for a table, please. Please, man. Don't do that. You draw that table, don't be lazy. Now, natural selection, the environment is a selective force. Like I said, those butterflies. And when you do a table, you must, if you say who's the selective force here, you must say who's the selective force on the other side. You can't now talk about something else here. You must talk about who's the selective force here. Humans are. And you can see that humans are now deciding here what to do. Then selection is in response to the suitability to the environment or environmental pressure, because nature is it. So that will be that mark there. Now you must talk about how selection is being done on this side. On this side, it's to satisfy human needs. Then they want to have desired characteristics. So that's the second one. And then the third, it occurs within species in nature, but humans actually were able now, not in this scenario, but in other scenarios, were able to interbreed different species. Now, we're clever like that. Right, so those are the 3. You could have had any 2. Right, explain 1 disadvantage of selecting breeding. Okay, selective breeding, always, the first thing, it will decrease your variation, your genetic variation. Right? Immediately, think of that. Because if you interbreed things, the genetic variation goes down, and that's never a good thing. Right? And your gene pool may increase the susceptibility to disease of the population. That's why animals that's purebred, and they interbreed them so much, they are very sickly, right? And they fall ill very easily, hip problems, all those things, because of interbreeding. Reduced wool in this case, because remember now they are breeding them to lose the wool on the stomach and the wherever, right? So now there's less wool for them to sell. Exposed skin, remember they are walking in the veld. Now those tummy is open and it's open there. So now it's less protected, because that's why they have wools, to protect them. And now they can get sunburn and insect bites and all other stuff. And like I said, genetic disorder due to interbreeding. Right, so fewer sheep survive. Again, explain. Explain is always 2 parts. Eh? Reduced genetic variation, that's the cause. What's the effect of that? Gene pool may increase susceptibility to disease. Can you see? Cause and effect, it's explained. Can't just say one thing. You must say the cause and how is it affecting, or how is it now doing whatever. Right, the how or the why. Right, punctuated equilibrium, you also struggle with this one. Okay, now I must go back. Oh, goodness. Now I won't be able to go back. How do I go back? Oopsie, now running ahead.
Dreams. What is your dream? Yini iphupho lakho? Not your parents' dreams, not your mom's dream for your life. What is your dream? Is it to get a bursary? Is it to study science? Is it to become a DJ? Is it to become a singer? What is your dream? Iphupho lakho. Once you have that dream, what I need you to do, I need you to write it down. Ingole mofati. Write it down, ibhale phansi, and read it each and every single day when you wake up. Read it each and every single day before you go to bed, and remind yourself of where you want to see yourself. What is your dream? Write it down, meditate on it, and go after it with everything that you have.
Right, so punctuated equilibrium. Just have a look at the difference between gradualism and punctuated. Gradualism is you start off with a common ancestor and gradually it changes through millions of years or thousands of years. You can see how it's gradually changing. These are all transitional fossils, right, or transitional species. It's in-betweens. They look a little bit like this one, but they also look a little bit like that one. Right, same on this side. Speciation happened there. So you can see the butterfly gradually changes. Now you have a total new species there. Right. With punctuated, nothing happens for thousands or millions of years. And all of a sudden, punk, I always say punk, punk, and there's a new species. Can you see there's no transitional to other species? It's not gradually happening. And all of a sudden you've got this new species there and it looks different. But you still got your original one and here's a new species on that side. Just suddenly punctuated, right? And they don't have transitional species. There's no transitional species here, but here is. But it's still natural selection. Don't think it's not natural selection. It's still happening through natural selection. It's all about the time here, right? It's fast, and then you have a new species. There's no gradually over a long period of time. Now you get a new species. Same here. You can just quickly look here. Gradually changing the beak, gradually getting longer. Transitional fossil. They're the same over here. But if you look at this one, there's the beak, and all of a sudden, whoops, there's the beak over a short space of time. The same here. There's no transitional fossil. There's no transitional species. This is not a true, this is just like a picture. It's not scientifically correct, but a horse, this one is correct. Gradually. It changed into bigger. Can you see? It was small, bigger, bigger, bigger. This is not the real one. It's just to show what the difference would be if it was punctuated. Smaller horse looked the same over a long period of time, and all of a sudden there was a big horse, a new species. Right. So this was gradually over millions of years. This was nothing happened, and all of a sudden you had a new species. Right. So what did go wrong in Gauteng paper 2 this year? The diagram represents a concept in evolution. Here you can see here's the common ancestor, and poops, all of a sudden there's a new species. No transitional fossils. A white mice, and the original one is still there, and here's speciation happened. Now there's a black one, right? A new species there. So no gradual transitional fossils. So identify the concept, it was punctuated, and give that definition. This is just the one that you get from your exam guideline. It's evolution involves longer periods of time where species do not change, nothing happens, or change gradually through natural selection. And this alternates with short periods of time where rapid change will happen through natural selection. Right, so that's where you went wrong there. But let's just look at human evolution as well. Human evolution, I'm not going to go through all of this because of time. I'll just say that you need to know what the similarities between us and the African apes is, right? This picture here shows it nicely. Also, what's the differences? Bipedalism, quadrupedalism, and what is the evidence they use for that? The foramen magnum, S-shaped spine, and the pelvic girdle, right? Cranium size, palate shape, dentition, prognathous or non-prognathous, and cranial and brow ridges. You need to know the differences there. Right, and I hope you know it by now because you were supposed to study for the prelims. Right, then human evolution also has 3 lines of evidence. Fossil, that's where the transitional, we look at the transitional species. And remember, what is a transitional species? It's got the characteristics common to the ancestor and to the descendant. Right, so that's a transitional. Genetic, they look at the mitochondrial DNA because it's simpler than the DNA in your nucleus, right? And mutations take much faster there, so they can actually trace back these mutations, and they did that, and they found that mitochondrial Eve lived in Africa 230 years ago, right? 1,000 years ago. Cultural evidence, and look at the tools that humans made, and that also relates to brain capacity. Right, then you need to know, people, you need to know the fossils of South Africa. Right, this is a very important. Africanus, Stonechild, Mrs. Place, Littlefoot, Karabo, Sediba, who found them? Right, it's very important. Naledi, where was it found? Who found it? Lee Berger, he found 2 of the big fossils. Homo sapiens, who found it and where? And remember, this is now very old Homo sapiens. It's not like we're going to the cemetery now and digging up graves. It's very old ones, fossilized. Right. Out of Africa hypothesis, what's important here? You must know what's the fossil evidence. These 2 were only found in Africa. That's a big thing, only found. And then this one, the oldest fossils were found in Africa, and the younger ones in the rest of the world. Right. Genetic evidence, mitochondrial DNA, trace the oldest female to East Africa, looking at those mutations. Right. Now, this question was asked, and this was just testing, did you study this? So the ones who did not study this, who didn't know your fossils, you couldn't answer this so well. Well, you couldn't answer it. Well, there was many answers you could give, because they were asking, select one Australopithecus fossil. Remember, there's many that you have learned about. You're supposed to know about Afarensis. You could have chosen this one. Then you would have said the common name is Lucy, and giants were found it. Or you could have said Prometheus. We don't actually talk about Prometheus. We talk about Africanus, Littlefoot, and there's the one, Ron Clark, who found it. Right. Or you could have said Africanus, Stonechild, Raymond Dart. Or you could have said Africanus, Mrs. Pless, Raymond Dart, Robert Broom, or Ron Clark. Right? Or you could say Sediba, Karabo, Lee Berger. So they gave you a lot of options there that you could have answered, yeah? It just, you needed to know it. State 2 advantages of a larger cranial capacity. Remember, cranium is this part of your skull where the brain is. So the bigger one shows a bigger brain. Right. So what is now the advantage of a bigger brain? You have a bigger brain, obviously, that's a larger cranial capacity. Speech, because we can talk to each other, we can communicate and hunt together and so on and survive. Higher intelligence, complex behavior, processing of information, that's how we were able to adapt and survive. And we could process a large amount of information. That's why you can study for your exams. Now they've asked, and this was a very difficult question. So I understand if you didn't get this, but it was actually not so difficult, it's just asked differently. How genetic evidence, now you must think, mitochondrial DNA, supports the hypothesis of ancestors of Chinese Homo erectus originated in Africa. Originated in Africa, and they went to China. That is out of Africa, people. That's what they're talking about. So, you talk about genetic, is they analyze the mutations of the mitochondrial DNA. There you already have 2 marks out of the 3. Right. So it was inherited only from the female. The oldest female ancestors were found from Africa. They were found in Africa. They traced these mutations back to the fossils they found in Africa. Right. And the Chinese and African Homo erectus fossils share similar mutations in mitochondrial DNA. And that's how they said, okay, no, but they are from there. Right, but I mean, if you have this 1, 2, and that one there, you had your 3 marks. Right, so it wasn't so difficult, but it's just a different way of asking it. Right, and then I am at the end of today's presentation. I hope that you listened and you learned from your mistakes. Please, please think about this when you study for your finals. Thank you very much, and good luck with your exams.